Hello and welcome to today's special panel discussion on mastering the complexity of CGT process development, how to meet challenges on the path to commercialization. Today you will hear from 4 experts from Miltenyi Biotec: Boris Engels, Daniel Jacobs, Silvio Weber, and Dr. Erman Bonenkamp. I'm going to be asking them a variety of questions connected with manufacturing, development, automation, and more, and then we'll wrap up with a live Q&A from the audience. Just before we get started, I'm going to offer a little bit of background on our experts. Boris Engels is Senior Scientific Director for Gene Engineered T-cell Therapies. He has expertise in cancer immunology, and he leads teams advancing CAR and TCR redirected T-cells for life-threatening diseases. Daniel Jacobs is Senior Manager, Cell Factory. He is responsible for the scale-up of the clinical manufacturing and the setup of commercial manufacturing of CAR-T products. Silvio Weber is Scientific Director, Industrial Workflow Development, Cellular Therapy. He leads the delivery of automated cell and gene therapy procedures using the Burr CliniMax Prodigy instrument platform. He also coordinates the development of customized processes for T-cell immunotherapy and stem cell engineering. And then finally, Dr. Erman Bonenkamp is VP Business Development, APAC. He has a strong background in CAR T-cell product development and global commercialization, and has driven success through strategic alliances. That's it for the introductions. So now we're going to get started with the main discussion. First of all, we're going to focus on scalability and consistency. So let's start with discussing the key challenges that small biotechs face in scaling up manufacturing processes for cell and gene therapy products. I'm not sure who would like to start with this one. Herman, perhaps we can come to you.
Yeah, thanks, Steph. I think it's a good question for basically setting the scene. So if you usually, for my experience at least, work with smaller biotech companies, the startup companies that let's say usually have a certain technology they develop that they would like to bring into the clinic. They would like to help patients as soon as possible, so time matters, and obviously budget is usually restricted. And manufacturing is one part of the avenue they need to do, but that's not the main focus basically, because they have developed technology in other spaces. So usually they would like to have something that works quickly and gets these things out of the way, basically. And so there are probably tons of different solutions, obviously, you can go for. But one, let's say, path has been shown to be working quite well. On one side, that you establish your own laboratory environment with the technology you'd like to use, and use a technology platform that has, let's say, a proven track record, so that you know, okay, this works somehow. And additionally, to work with, let's say, CDMO companies that have a certain track record in the field and can help you with the development, so that you really don't lose time in the beginning and spend your money and time wisely. So this is, I think, some of the learnings we had in the past that is working also quite successfully in the field. Yeah, so that this manufacturing parts are really getting out of the way. But ultimately, that's part of the discussion today. You need to deal with this anyway. Yeah, so and that's a critical point, that from the beginning you take this seriously, and you try to find solutions in the context that work also long term.
And then next then, how do we ensure scalability and consistency in manufacturing processes for CGT products, especially when transitioning from small scale to commercial scale production? Silvio, do you want to start with this? You're on mute.
I was really setting nicely the scene for this question. And from the R&D perspective or development perspective, I think there are 2 major things. So first is really trying to have a stable ecosystem. Yeah? To set something, if it comes to people, machines, materials, but also methods, that is robust as possible. So for the people, it's very important that they are well trained and that they understand the full life cycle of pharmaceutical development, from preclinical to commercial, in order to take the right decisions as a cross-functional team with many others. And then the machines, as Hermann also nicely said, the more they are pre-set up and defined and have a platform approach, and the more they are automated, the easier it gets to develop your drug. On the material side, also, I think maybe Boris will touch a little bit on that. It's key to have them GMP grade from the beginning, but what does it really mean? And then the methods, well, that is closely connected usually to the machines, especially as we are now moving more towards software-based systems and platforms. So we try to fully integrate them, and that's a key thing. And this leads me to the second point, which is then standardization. So this comes on top. It's really getting all of these puzzle pieces together to one standardized workflow that is then mirrored by manufacturing batch record. And I think Daniel will talk a lot about that later, because he's really from that clinical, commercial, real-world environment where we from the R&D are more trying to bring the best to the entire game.
Maybe just to add, so thanks, Silvio. I think from the R&D perspective, when you develop the processes first at small scale, it's really important to have, I think, the large scale commercial setting in mind, because there are many things that you can put on the right path early, having that process in mind. Even if it may be a little bit more complicated at first, but I think having that final vision commercial setting in mind is really helpful. And that pertains to the process, obviously, but also the reagents you use. So if you try to work with reagents that are either GMP grade or available in GMP grade early on, this will alleviate many headaches later on when you have a beautiful car that works in animals, but then need to move that into a clinical or even commercial setting, and you have to put in changes and tweaks. So I think having that final picture in mind is really important.
Great. And then next, what are the critical factors to consider when designing manufacturing processes to accommodate increased production volumes without compromising product quality? Quite a complex question here. Who wants to take this one? I think you're muted, Silvio.
Yeah, I think I can take it for the beginning. So again, standardization of workflows is key. And one thing we usually forget when we talk about processes and manufacturing are the analytics. But they are really key, because if you want to understand your product, you have to define so-called critical quality attributes. And you need really reliable and robust assays for that. Because during the life cycle of the pharmaceutical development chain, you will very likely have changes. In order to implement them and to ensure comparability, this will be a key thing. And then the second thing that I think is also very important, we from the R&D try to understand this real-world environment in the clinical commercial space. So I think they always try to have something like lean manufacturing principles. It's not only the process itself, but you have to attach reagents to the process. You have to bring in things to the manufacturing environment and people. And this goes beyond that, what we usually do in the R&D. So we have to revisit these processes continuously together, also with the MSAT and clinical colleagues.
And if I may, to add as well, I think what is important, and I think Silvio touched on it, is the really not focusing only on the process, but also the analytics. So to have those in mind early. What helps in the scaling up is obviously automation, both of the process as well as the analytics. If you think, for example, of multiplexing of the analytics to run several at the same time, is extremely helpful. One, I think, example where upscaling has been difficult is if you have, for example, long ways in between. And the process development, building, and the analytics. So if the samples have to travel, I think those are early considerations as well. And then lastly, I think one aspect that Miltenyi has been pushing forward and quite successful is the concept of a ballroom, where you have a single GMP facility where you have closed devices and you can generate several products at the same time. I think all of that really helps with the upscaling.
Yeah, perhaps, Boris, that's a good point. I think that brings me also back to one of these initial pain points: budget. Yeah, so ultimately, there's always a limited budget if it comes to outscaling, and having let's say limited spaces available. If you really like to then bring a certain amount of cell therapies to market, that helps. Yeah, it helps to get costs down. It helps to reduce also the needs of highly trained personnel in the context. And it makes also it easier to tech transfer these processes. Not that the tech transfer per se is easy, but it helps to accommodate this much easier if you think about licensing this out, bring this to another country, and so on. So I think these automation and smaller footprints of facilities are key aspects that someone should consider in the context.
And Daniel, I'm gonna come to you for this next question. Have you encountered any specific challenges in maintaining consistency in product quality and yield during scale up? And importantly, how did you address them?
Yeah, for this, I think I need to pick up what Boris said. We're using here ballroom design at Miltenyi, where we use the platform, the Prodigy, and this really helps because in the end, you basically do not really scale up, but you multiply. You use more Prodigies. So then in the end, it becomes an issue of timing. So if you do certain processing steps, you need to be aware of the timing of the different steps. Because some of the equipment you will not be able to multiply. For example, biosafety cabinets, you will have a certain amount of those, and you need to keep that in mind when scaling up. And obviously open handling steps. So if you could reduce them as much as possible, this all helps in scaling up. And since we use a closed system platform, we did not have that many issues here. So I think this is a good approach to go in this direction.
Great. Some really great insights so far. But we're gonna switch track a little now to focus more on process development and optimization. We're going to delve a little bit more into these areas. So Silvio, I'm going to come to you for the next question. What methodologies do you employ for process development and optimization in CGT manufacturing?
Yeah, Stephanie, let me answer that. Maybe there are two different worlds. I would say there's an ideal world and there's a more realistic world, especially in the field of autologous ITMPs, where we have very complex nature of the product. So in an ideal world, I mean, you would have a so-called quality target product profile that defines your product. This is then characterized by so-called these critical quality attributes. And once you have them, you do a risk assessment and you try to find the design space in where your drug product is working well. So this needs a lot of analytics and control. And especially in the beginning, I think this is something, if you don't have a lot of historic knowledge and also already good understanding, it will be quite difficult. So it will be most likely a little bit more empirical and trying to change variables one at a time. But what really helps here is if you use such an automated platform on manufacturing, also analytics, to keep the variables as small as possible and really just check if you change from one to the other variable. So that is something that is really more setting up a robust process. But well, in the future, I think if we start to implement more something that we call process analytical technologies, so having inline measurements, for example, continuous inline measurements with small volumes, because that's often critical. You don't want to waste too much of your materials, of your cells, so you have to move to more microfluidic systems and recycling maybe cells. So this is probably the future. And then we have a better way to continuously control our critical quality attributes, and by that really to optimize this process development tools. And then also implement design of experiments, multivariate analysis, as we are used, for example, from biologics.
And then perhaps it would be really nice if we could talk a little bit more about transferring the process from R&D to the GMP environment and how exactly that's done.
We can take a stab there. I think similar to what I mentioned earlier is kind of having the final process in mind. Along those lines, I think it's also important to work together early with the manufacturing facility, the GMP specialists, so that you don't implement aspects that will be later on very difficult to be used. It could be certain processes that require specific hoods or specific reagents or instruments which are not GMP compliant. So I think having that dialogue really early is crucial. Similarly, if for the process development, if you have some sort of a checklist of what aspects need to be assessed, what needs to be done, that will then you can work through. That checklist could be worked out together with the manufacturing group, so that you know exactly what is needed, and then the transfer would be easier that way.
And of course, whenever we're talking about manufacturing, any kind of therapy, a topic that is becoming increasingly important for the industry is automation and technology. So what role does automation play in CGT manufacturing? How do you leverage automation technologies to improve efficiency, reduce costs, and minimize variability? Who wants to have a stab with this one?
Yeah, I think I can go ahead. I think it's obvious, right? I mean, automation is the best friend also of standardization. That's a topic we touched on before. And so especially if you combine machine automation with a digital integration, this will definitely help you to save costs on many ends. So already in the development situation, because you can do less runs, because you don't have to control so many variables. But then also later on in the clinical and commercial space, and I think then Daniel will maybe touch on that, it will help you to drive down failures during manufacturing a lot. But one thing, and we talked, for example, about open handling steps, is we often forget that automation is not only the process itself, but all the things that go into it. And so we also have to think about automating these processes when we transfer samples. Boris described that, right, from the manufacturing to the automated analytics, for example. So how can we do that? And is that built in by design if we use our, for example, tubing sets, disposables, that we put in certain kind of sampling tubes that we can maybe bring to our automated analyzer, things like that. So this will help us to really keep the chain of custody, chain of identity, integrity, and that is also key here for automation. Another aspect is also very important on the documentation side. So you surely want to have something like an electronic batch record that also then integrates to the entire patient journey and not only the product journey. So maybe, Boris, you have some further thoughts on that.
Yeah, I think you touched on most of it, but I think indeed it's automation of the different steps. So not only the process itself, the handling of the product, but also the sampling steps, for example. So if you automate such steps of mixing the cells, taking the aliquot for the sampling. If all of that doesn't require long standard procedures for the staff, but rather is automated and repeatedly done the same way, is really helpful to control the process. And obviously that makes the costs lower as well if you don't have each product being handled by a single person throughout, but rather it's supervising what is ongoing in parallel in several machines. I think that helps with regard to the costs.
Yeah, I agree, Boris. I mean, if I may shortly add, I think you said it's thinking at scale is key, especially at the beginning. And here, for example, if we think about media preparation, so if you just have a couple of runs a week or a month, it's totally different from 20 or 30. So maybe then you also start to automate this process more and do something like a bulk media filling or things like that, and it can also be automated. So this is really also depending on the stage of development, I think, when you start to bring in automation. I can give you another example. So you have things like dose escalation studies in the beginning of clinical development. So do you really want to develop a fully automated system for a couple of runs that you maybe have to do there once you have decided for a certain dose? Well, that is some investment. I think Hermann explained about all the advantages that you have if you automate. But if you have a very small sample size, that sometimes it's even better to stay with a certain manual way of doing the process than bringing everything to a fully automated approach. So it's also a little bit of a balanced decision, I would say, here from here and there.
Yeah, perhaps I would like to add 2 additional points. But we usually, if clients are rather new to the field, are having, let's say, some issues in the beginning. The first thing is control. If I have these boxes that automate all my unit operations, I may lose control. But ultimately, as you can tell exactly the machine what to do in a certain sequence of time and what exactly is happening, you don't lose actually control. It's just that the instrument is doing what you tell the instrument. Yeah, so it's from that point not really a loss of control. And as you can sample what you need, there's not really a loss of control. It's actually in a lot of senses better because you don't need to do a lot of in-process controls as you usually do if you swap 2 different instrumentations in this specific time. The second point is cost. Yes, of course, you have some investment costs in the context, but ultimately you reduce your requirements of staff on controls, on variability so dramatically that it pays off immediately, basically. From these point of 2 different argumentations, it's an important, let's say, these are important aspects. But usually, if we discuss this in detail, we can then also argue very good points for automation in this field, basically.
Yeah, also looking at this from a manufacturing point of view, especially when you move into commercial manufacturing. To reduce risk is always key. I think the CGT manufacturing process itself is quite demanding, also in regards to training of personnel and things like this. And to exclude certain failure modes is very much appreciated. So automation really helps us in the scale up. It reduces also cost, as my predecessor already said. Yeah, so good thing to do.
And then, have any of you encountered any challenges or limitations in adopting automation technologies? And if so, how did you overcome them?
Yeah, obviously, you will always have borders on what you can do, and every time you get close to this, you will face issues. So, for example, if the dose needs to be very low, you cannot do an automated formulation, which is one of the most critical points during manufacturing. So then you would need to switch to a manual formulation, which is a lot harder to achieve. And for example, moving to the other direction, if you want to manufacture a big amount of cells, then at some point culture chambers might be too small, and then you need to think about ways to scale up there, or you need to multiply your platform.
Maybe, yeah, I agree, right? With automation, you gain control of certain aspects, but then obviously it also sets boundaries. So, and we see that every day where we think we have thought of all possible situations, but then yet again you hit some limits. So I think it's a continuous process in optimizing and allowing for those sides, those special cases to be adapted as well.
And linked in with technology, I guess, is infrastructure and facilities themselves. So tell me, what are the key considerations in designing, manufacturing facilities for cell and gene, including layout, equipment selection, infrastructure requirements? What do we have to think about there?
I think there goes a lot of thought into this. So first of all, you need to ensure EHS and biosafety. That's a given, and this needs to be implemented. And then I guess the next question is, what are your capacity needs? You need to define this first, and then the key is to define the material, personnel, and waste flows, so that you clearly define them and size them for the intended use. You need to think of how to replenish what you need inside of the facility, and then so this is like general thoughts that you need to start with, and then it gets more specific on the product, because there's a couple of product specific requirements that you need to address. You need to look into cross contamination. So there's a difference if it's a one product facility or if it's a multi-product facility, so you need to adapt there. And in regards to cross-contamination, you need to look into your biosafety cabinets. How do you handle the exhaust air, for example? How do you prevent mix-ups in the whole process? Then obviously, there's a lot of changes in the CGT world, so you want to be versatile. So you need to think about, can I have versatile equipment that I can also use for other purposes later on, maybe. And then if you do, also during the design process, you need to think about business continuity. Because if you just start this thought process after you design your facilities, it's too late. So, for example, you need to look into which equipment doesn't need an uninterrupted power supply and stuff like this. And then also digitalization needs to be taken into consideration, not only in regards to monitoring, but also how do you ensure GMP documentation.
Yeah, I think generally it's a very interesting question. So Miltenyi has done around 15 projects with external clients on designing their facilities, and Daniel had, I think, a very comprehensive list. Okay, what do you need to take into consideration? But usually we ask the first question to clients, okay, what do you want to really do? Yeah, and usually they don't know in the beginning. So that is really an initial journey on the user requirements. What do you want to do and how do you implement this? You want to have a purpose-built facility versus maximum flexibility. And that obviously is not always working best in the context. So you need to find a balance here to keep also obviously your budget under control. But I find this usually quite interesting if we work with CDMOs that build a CDMO business around certain technologies, and we help them to understand what they want to do and design then accordingly the different rooms, flows, safety, risk mitigations, and then in the end they say, oh, we have everything done, and then half of the facility is missing because we didn't talk about QC. So that's obviously then a big consideration that always needs to take into. You really need to think about what do we really need in the end, and do I like to do it in-house or external, and I need to have a lot of capabilities for CGT. So we talked there easy about 8 to 10 assays that need to be run if you talk about a CGT product. So that's also a big learning for us.
That was exactly the example, right, where I heard that the facility or the building for the production and for the QC were kind of across the campus, which makes obviously the process quite difficult.
Okay. And then, Daniel, we came to you before to ask about, challenges related to automation. So I'm going to come back to you for this one as well. What about challenges related to facility design or infrastructure limitations? Obviously, discussed a few of them already, but, are there any others you can talk about that you've faced and talk about how you've addressed them?
Yeah, so first thing that comes to mind is parallel processing. As I already said before, you have a certain amount of equipment that you will not, that you will only have a finite number of. So this is something that you need to look into. And then also multi-site manufacturing might be an issue. So if you manufacture a different site, but the same product, you need to ensure comparability. So if then the design is differently, this might result into issues. Biosafety is a very important point here that is always challenging. And so overall, when you have those issues, you always need to look into your special situation to find solutions. I don't think there's a one-size-fits-all solutions for those challenges. Also because this CGT field is evolving so quickly that you always need to look into your special need at that time, exactly what Hermann said before.
I think we've covered a lot of interesting topics so far, technology, optimization, lots of different areas, but something we haven't really touched upon at all is supply chain management. Obviously this is really, really important. So I want to ask, what do you think are the critical components of the supply chain, for CGT products, and how do you manage sourcing, procurement, and inventory management?
Yeah, I think for our process, serum, obviously vector, final formulation, and cryosupplement are the most critical products. Here at Miltenyi, I'm in a very nice situation from a manufacturing point of view because we basically manufacture most of those things ourselves. But in general, key is always forecast. Yeah, you need to build up good relationships to your suppliers, and you will build the relationship best if you supply them with a good forecast. Then obviously you need to implement risk mitigation steps like second sourcing. Yeah, I think these are general considerations that you need to look into. And if possible, like we do manufacturing everything by ourselves, or the other way around, if you're an external customer, we will provide you with basically everything you need for your manufacturing process.
Yeah, perhaps from the commercial point, it first of all looks like a very boring topic. If you think about it, we have this great asset, we want to bring this into the clinic, we want to help patients, and then we just order our stuff and then we manufacture. But ultimately, as in the last decade, hundreds of companies are moved into this field. Several companies moved to commercial. 10,000 of products are nowadays manufactured. Within the next 5 years, the scale out to 100,000 of patients, companies have announced. So obviously, making such materials and getting very accurate forecasts, especially if it comes to higher demands, is very crucial. So this is really one of these important steps where we also proactively go to clients and say, now is a good time to talk about your needs, actually. Not that, anything is binding or whatever. It's just that we need to know to understand what we need to manufacture next year. Yeah, do we also from Miltenyi's side need to build up a new plant for making media, cytokines, and so on. So this is a very important step in the context. And another one, I just can mirror what Daniel is saying, lentiviral vector supply is usually one of the pain points, let's say, and adds to time very early on in certain projects. Yeah, to get the vectors in time into the project. And that's, I think, also important to either do this properly in-house or outsource this to suppliers that have a long-standing track record in the context, and then develop proper timelines in the context. Yeah, and this is, I think, a very important step. Before you think about manufacturing a CGT, think about, okay, I need a vector. Okay, how do I do this? And then develop timelines, and then manufacturing comes then accordingly. Yeah, but that's, I think, also a very important topic that should be dealt with right from the beginning.
And then what about any other critical parameters for managing cost of goods, for manufacturing and quality control? Is there anything you want to say on how the highest efficiency gains can be expected, or other key parameters to look at in the supply chain?
Yeah, I think if you start developing a process, I think you need to have certain flexibility to let's say develop it properly. If you later on in a commercial stage have a very good understanding how your product looks like, how the manufacturing looks like, then you can very detailed look into efficiencies. I may need not a 2-liter bag, but a 1-liter bag. I may need certain cytokines in different formats and so on. If it comes to production of 10,000 products, then it's worthwhile to look in each of the steps to get all the efficiencies out. Yeah, to make custom products, yeah, and to get this to a much better price in this context. Yeah, it's not worthwhile if you think about, I make now my first clinical trial, 10 patients, to look into all these details, but it's important to have a vendor with whom you can look into it and get then all the efficiencies out. And ultimately, it's also about how's the global network looking like? Can I supply this to all the countries I go for the manufacturing? Do I get all the regulatory support for all the countries I would like to file? How does the distribution looks like in the context? How's my inventory management looking like versus of the supplier? Can I get help there in the context? So a lot of detailed in the end where the supply people need to look into and make really proper plans in the context, how to get the efficiencies out and get all the needs what you have then in the context, if you think about commercial manufacturing.
Great. We're coming to the end of the discussion now, but before we wrap up, I think it's really important to look at compliance and quality assurance. So, how do you ensure compliance with regulatory requirements, quality standards throughout the whole manufacturing process?
Yeah, maybe a bit counterintuitive that I picked this up first as an R&D guy. But I think this is something we learned during the design of processes. It's absolutely important that the employees at least basically understand all these different aspects of the regulatory and quality landscape. And the idea is not, to become QA or RA experts themselves, but rather to understand when to work as a team together with the colleagues from QA and RA. And ideally, you already have a standardized business or process, that you start to bring in these kind of people to your development situations that you have. And this will help you to make sure that you will align with these kind of requirements and then deliver something to the MSAT or clinical production colleagues that is already fit for purpose. And maybe last but not least, if it goes more to the business side, I know that Hermann is more the expert, but in the end, it's the client's product or the product that you're looking at, which needs certain quality attributes. So you also have to make sure that QA and RA are not oversteering things. Yeah, sometimes they're really into their stuff and they're really keen on doing the best they could. But here it's really trying to find the right balance all together as a team. But if you do this right, then it's really a game changer, because the quality in the end is what sets also your performance and also the cost of good aspects in the end. Because a failed batch is that what is most expensive, far more expensive than being maybe not perfectly lean or so.
Yeah, and then the manufacturing, you obviously follow GMP guidelines. You make sure that you do all the quality controls and then adhere to your SOPs. It's not rocket science in the end, as long as you define your processes in a good way.
And then what about quality control measures and analytical testing methods? What can you tell us about how those can be employed to monitor product quality and safety?
Yeah, obviously you want to monitor the process as close as possible. So you have expectations on where you want to be at a certain stage during your process, and we measure cell count, viability, also potency, and then you also need obviously your safety measures. You need to make sure that identity is correct, but also vector copy number. You need to ensure sterility and contaminations. And last but not least, we obviously do a visual inspection at the end of our manufacturing process to make sure that everything is in the right order.
Maybe I can add to that point, because indeed it's a longer list of assays, right, that Daniel mentioned, and many of those are kicked off in R&D, because we use them to characterize the cars that we manufacture for the preclinical development. And there actually comes a point that Silvio made, is that if we are aware of regulatory requirements in R&D, we can already start using or designing these assays fit for purpose, and not maybe developing a 16-color flow panel that is scientifically really, really interesting, but then for a commercial setting maybe overdoing. So I think that, coming back to what I mentioned earlier, that the really thinking of the final process and aligning early is helpful in designing these assays as needed, and really pinpointing what they need to show, and that is key, and then also sufficient, and design these appropriately.
Yeah, perhaps to add to this, what we also can observe is that during the clinical phase, obviously many more questions people like to ask, R&D questions, clinical questions. How do we get a match on the cells we do and on the clinical outcome? So many more assays are done during this phase that drives more insights that are not relevant for the manufacturing and quality in the end, but are from the scientific point of view quite interesting. Yeah, what kind of cell do I have? Is a young cell a more terminal differentiated one? What is the effect in the end of outcome? So I think these are very interesting questions, obviously, but a lot of these, let's say 16 or 35 color panels, are not in the end something that you want to do in any manufacturing for commercial setting, because it doesn't give any additional insights into the QC, but is important from the scientific point of view, obviously.
At this point, we're starting to run out of time, so we're gonna jump ahead to the live Q&A session. For everyone watching this live, please submit any questions you might have using the box on your screens, and we'll get through as many as we can in the time that we have left. So the first question for our Q&A then: what do you undertake to validate the process?
On this end, I would say we support. I mean, we are very much involved in these engineering runs, where we really set up together the manufacturing batch records with our colleagues from the clinical production side. But as more as we are moving towards validation, the more the clinical guys take over. And I think there's a solid approach, right? How you validate the process and also the analytical assays. So maybe you can talk a little bit about the pain points you are having, and the back and forth that you also have with us in the R&D in that aspect.
I think it's important to have the critical quality attributes in place, and then you can design a validation plan on how to look into those. And then you will follow up. I mean, after we did the engineering runs by Silvio's team, we would then follow up by doing validation runs in the facility in which we would later on also manufacture.
To be honest, when the first time I saw this validation master plan, how this is called, it was, I think, the biggest table I've ever seen and most comprehensive one. So I thought, okay, this would have been nice in my more scientific academic career to have something like a to Z plan. And that was very fascinating for me to see that really everything is planned and looked at and is like a leading document to guide you through this important process. Because what you validate there, right, that is the cornerstone of that, what you're doing later on in your also commercial manufacturing process. So it has to be comprehensive and very exact. And then also goes together with a project plan, another document that was really out of my imagination when I saw it, with all these different lines and the interdependencies coming together. So they're also digital, I would say, integration is key here to understand where you are going. So at least for I was more an observer, but I was really fascinated, to be honest.
And I think the third big document that you need to generate is a risk analysis that looks into all the different aspects of the process. But out of this, you then generate the critical quality attributes, and then you can design the validation plan accordingly to make sure that everything is really reproducible in the end.
Next question then. How do you balance the need for improvements of process robustness and efficiency while maintaining product specifications or regulatory requirements?
Maybe I can take a first stab at that. I think what is important is what Daniel just mentioned, right? The critical quality attributes. So and the risk assessment. So I think in the cell and gene therapy space, as it is still relatively young and growing, there is a constant improvement of the processes and a constant improvement of the processes even in the commercial space, but during clinical phase, absolutely. And what is really important there is to have those CQAs defined and then do a risk assessment of your process improvements, how they may or may not change those. And at times, I think there's different levels of improvements and of changes that can be implemented by different levels of workload. So it starts off probably with small changes where the risk of impact on the drug product are low. So there possibly a risk assessment is sufficient. The next step would be to have comparability runs to show that your process produces the same product. And then if indeed you do bigger changes, you may be challenged with showing clinical efficacy on the far end. And maybe one example where such a process improvement has indeed even led to a new product is if we look at Kite and Tecartus, where they have improved or have changed the enrichment of the T cells that now led to a whole new product. So I think there's a broad spectrum of improvements and how then they can be implemented.
And then have you encountered any supply chain disruptions or shortages that have impacted CGT production? And if so, how have these been mitigated?
Yeah, maybe I can take or say something to this. So I think it comes back to the supply chain part. Yeah, so I think it's very important at the beginning, if you start such projects, to align with your vendor base, with your vendors, what you want to do, and especially when it comes to higher numbers, that there's a clear alignment. Yeah, so we only can obviously provide what we know, and that's, I think, a very important part to have a good understanding what's needed and when. Yeah, so how does your plan look like? And it's not about one product to deliver. Yeah, it's really about to guide this in this whole project plan in the context. And there are obviously more critical products than others. Yeah, and as already pointed out, when it comes to lentiviral vectors, for instance, this is one of the key aspects that needs to be taken care of in the very beginning. Yeah, so that's also, let's say, from the risk part of point of view, identify the materials that are really, really critical and take care of those. That's, I think, an important aspect.
Great. We are very close to running out of time, but we're just going to try and get through 2 more questions. So the first of these then, how do you balance the need for specialized expertise in areas such as cell culture, gene editing, and analytical techniques with broader manufacturing competencies?
Yeah, so we try to automate as much as possible. So in regards to manufacturing, we have the Prodigy platform that automates the manufacturing process as much. And right now we are in development also for a lab automation system that helps us to automate the processes there, which would be a good next step. And just in our facility right now, at this point, we are only in preparation for commercial manufacturing, so we are not yet doing this. So right now we employ more people on a more expert level because the processes are still a little bit to be shaped. And the more you move to commercial manufacturing, the more you will need to look into standardization, and you need then also broader manufacturing competencies rather than right now where you need to be more of an expert to do this. And overall, what we did is we tried to now to hire at least at the leadership level people with this background of commercial manufacturing to help us shape all of that, because CGT is still young. So there's not something that you can just copy and paste, but you need to develop your own processes depending on the products that you manufacture and on the equipment that you use.
So if I may very shortly add, what I really like if people are still able to have both hats on their head, so that they can think more in an expert manner, but also in a more generic manner. Because from time to time, you will have changes, even in the commercial setting. I think this was said before. And if you're completely inert against these kind of changes, it will not help. So sure, there will be more orientation towards a certain path, but a little bit fluidity or plasticity, I think, can definitely help all of us.
Then we have time just for one last question then. And I think this one's interesting. It's a little bit different to what we've talked about before, but you did just mention that the field is quite new. So the question is, have you implemented any initiatives to attract and retain talent in the field of CGT manufacturing, such as internships, apprenticeships, or professional development programs? Obviously the field is so new, so there isn't always that much talent around. So it is really great, to hear about how companies are inspiring the next generation.
Yeah, so internships is a bit hard because you need quite a bit of time to get into it, and then this really doesn't match with the usually shorter time frame of an internship. But we are part of the apprenticeship program here at Miltenyi, so this is something that we really like, because basically it's really hard to get employees that already have everything that we would need. And also there's not just one simple CV that we are interested. So in the end, we set up our own training programs, and also what we're doing right now, especially to attain or retain the talent, is we start with development programs, because I think there's quite a need for personnel. And since you cannot get a lot of them on the market, we need to develop those people and those talents on our own, and this is something that we set up.
And if we still have a moment, I think what is really nice here at Miltenyi is that we have the possibility to cross-train as well. So we have employees in R&D that are interested in gaining insight into how the cell factory GMP manufacturing works, and there's that possibility. So I think having both arms close together is attractive and opens up learnings across the company here.
in a way, it works both ways, right? So I think people would be that know, like with the hands-on experience on what they need to do, are highly appreciated in an R&D environment as well. So this is really a good setting here.
Great. I want to thank all of you for answering all of those questions. I know there were a lot of them. So thank you very much for sharing all of your insight and expertise. That is all we have time for today. If your question hasn't been answered live here in the Q&A, then don't worry, because we will pass over any remaining questions to our speakers so that they can answer via email. And if you wish to revisit the topics covered today, then this webinar will be available on demand in the next couple of days. Thank you again to all of our panelists, Boris, Daniel, Silvio, and Erman. And thank you to everyone for attending.
Thank you too, Stephanie.
Thank you. Thanks for having us.